Design is that I want your groups to explain the figures, and I'd love the rest of the class to engage in questions, things that are either unclear from their explanations or things that are confusing to you. My goal is by the end of the class today that people will have full understanding of this paper and the figures involved in it, I guess. Okay? All right, take it.
Okay, so for A and B of figure 1, the blue is showing the nucleosome phasing of like their research using the Solexa technique, and the red was nucleosome phasing found using other techniques previously. And the main difference between A and B, they're just showing different genes that they're looking at.
So for C and D, they're pretty closely related because it's the same gene, the NBPF10 gene, and they're looking at where nucleosomes are found. So they're using the Selexia sequencing tags to find nucleosomes at like 0.1, 0.2, 0.3, and so on. And then that's related to D because they use the electrophoresis gel to measure where that nucleosome is, and like on 1 it's about 140 base pairs, for 2 it's like 115, for 3 it's like 120, and that's just showing where the nucleosome is condensed and supercoiled, and that's why it's showing up heavier, because the base pairs are compacted together. That's what I got.
So this was important data to include because it shows that the Solexa technique is just as good and better than other techniques. They are able to see that previous research where nucleosomes are found and then where all of their nucleosomes are found.
I think that pretty much, we just open up the questions now.
Sure.
Sort of. Any questions about the figure? So like, this is one of like the questions obviously in the paper, but like she said, the biggest thing that they were, why this was even included is because they wanted to show how much more effective their research techniques were than previous. Because as you can see, like they not only found that their nucleosomes lined up with the previous, but they also found more. So that was the biggest thing about this graph or figure.
Okay, so it's like the difference between the B and C, just that it's at different spots. Like is C just more general? Because it says it's on a gene body.
There are 2 different genes that they're looking at for C or B and C. For B, they're looking at gene BRCA1, and then they're also looking at NBR2, and where the nucleosomes are found on that gene. And then for C, they're looking at the NBPF10 gene. So they're looking at 2 different genes.
The other note that I put into the explanation would be that they talk about the paper at C, so those B, A, and B are talking about mostly promoter regions, whereas C is an energetic region. So I think that they wanted to prove that they could do this technique both in a promoter region as well as an energetic region. So they're trying to prove that their technique has wide applicability across the
Is the energetic region, is that just like the middle of the gene?
not the middle of the gene, but
Middle of the
region
sequence.
between genes.
Oh.
So you can see that little box, that small shaded box is NPF10, and 1, 2, 3, 4, 5 is far, far away from that gene. So, so they are showing that they can do this not only next to genes, but in these areas where there are no genes at all.
I had a question about the scoring function, like that part in like the experimental procedures. So it said that they had like a 10 base pair window, they ran it along and they measured how many genes mapped to like the antisense and the sense string. Can you explain that?
So yeah, so the sense antisense is more about figure 2. So basically what you're seeing is these big peaks, the big black peaks that you see, is how many times they observe that sequence coming off of out of their reads, of their sequence reads. So if they saw the sequences, they would map it, And the lack of seeing that shows that that DNA sequence was not associated with the nucleosome. So the way that the methods go about this is they isolate the mononucleosomes, right? And then they identify the DNA that is found in those mononucleosomes and then sequence them and then figure out where those sequences are at. Right, so I think we've talked about this before, but the population, so if there's a population of cells, you can see that the vast majority of cells, you see the nucleosomes positioned, and that's what these histograms are looking at. Where do we see them most commonly? That helps. All right, any other questions for figure one? All right, figure two.
Let's take it. We probably should have figured that out.
Okay, so this is our figure. I'm gonna explain A. So that's up here in this corner. So the gray dots is where they sequenced all this stuff, and then this whole strand DNA, and the gray dots are where they found localized nucleosomes on basically like every strand. I think Madison might draw something on the board, I'm not sure though, just kidding, I'm sorry. And then the peaks are right around the TSS, which is awesome, because that shows that, because at the TSS, that's where like transcription starts, it's the transcription starting site. And you can tell right after this one gray dot, which is a +1, is right after the TSS, which shows that it's an active gene and it's being expressed, if that makes sense. Anybody else?
Okay, so for figure B, it's basically the same what she said, except for there's only one oval dot, so that's the only spot where they found the nucleosomes in a line. And the rest of it, they're all scattered around, so not in a line. And then they don't go, the little valleys don't go up as high because there's not as many found. And yep, Allie's showing a picture now. And then, so this is like unexpressed because they are all scattered around.
So that's like the gray dot, is because there is stuff in every line.
So this is where the gray dot would be, they're all in a line, and then the little scattered points are where there's not a gray dot.
And this is all the static, because there's no set pattern, everything's just very delocalized. So they didn't find a nucleus, well, they didn't mark a nucleosome there still could be a nucleosome on the strand, but there wasn't a pattern, so then they were like, nah. Next person.
Okay, so C is just using another technique called the chip sequence. So they're comparing an older technique that was used previously to their technique, and they found that the chip sequence technique wasn't as good because, so it measured the histone levels, and as you can see, the graph isn't as clear cut as the other one because there's just the one single peak, and the other one shows like 8 different nucleosome peaks, and this one only shows a couple. So they're saying it's not as good, especially at high resolution. They showed another graph on the other supplementary figures where it was in the lower resolution, the Celexa technique and the chip sequence seemed to match up pretty well, but then when the chip sequence was at a higher resolution, it didn't match up quite as well.
Okay, I'm gonna attempt to try D. So it basically is comparing the nucleosome levels
kind of like what we see up here in figure A and B, and then the Pol2 or the polymerase. And so we can see that we're looking for expressed genes are going to be the red and the green lines for expressed. And we can see that the polymerase is found at the start site right here, and then we see that the nucleosomes are found just off to the right of the polymerase, and that's because polymerase is needed to actively transcribe the genes, and so they're being expressed, they're being transcribed, so that's why it's good that we see the polymerase where we see it and the nucleosomes where we see it. But if you look at the non-expressed nucleosomes of the polymerase, they're both right about on the transcription start site, and this is because the genes are not being expressed, and so the nucleosomes are able to outcompete the polymerase for the start site, and so that's why we see them there.
Any questions?
What's so confusing for people
For C, why do they have the histone level rather than the nucleosome level?
I think it's kind of dependent on the type of test that they used. So with the CHIP sequence, they used the histone 3 antibody to identify the different histones. So it just was looking for something different, and this Alexa technique just did the nucleosomes. Is that accurate?
I think the difference here in their technique is that they were able to get to mononucleosome resolution, right? Whereas this, I think that the chip sequence is just not able to get to that level, right? So,
Because the chip still reads part of the linker region as well.
and then some of the fragments are larger than a.
Yeah, and they're not the same, so that's not good. We don't like that.
So like the MNase technique takes out
Oh, yes.
takes out all of this DNA, and so you're just left with the nucleosomes, and then the other one, it takes out some of it, and then you're still left with a lot of extra DNA.
Other questions? All right, 33.
Okay, so we're gonna start with B. You can see the different nucleosome levels. So for elongating, which was for active genes, when it allowed it to transcribe, the nucleosome positioning was in the right place, which allowed the polymerase to bind. But then over by the transcription start site. And then if you look down at C for stalled, the nucleosome levels near the start site were too close or over the top, so the polymerase couldn't bind that well, and then transcription couldn't occur. But then when you look where there's no polymerase binding, the nucleosomes levels just kind of run straight through. And we actually looked at the wrong graph when we were discussing, so we did not get to talk about A real fast while we were pulling ourselves together.
But I can try
Yes,
to do a
but
little-
Jessica, give you a little description.
So I think basically what it's saying for the elongated pole 2, just that it had a wide range of expression, but the with no pole 2 there wasn't very much because it wasn't present, it wasn't like bound to it and actively transcribing. It's, yeah, I don't know if that makes sense.
And again, especially in like the no pole 2 and the elongating where there's like very flat periods of just like little squiggles, that's not saying that there's not any nucleosomes there, just that they're not in the same spot across a whole bunch of genes, that their spot varies a lot.
take questions for.
So do you think like the stalled pole two graph is significant at all too? Or why would there be some expression still, but not as much as elongated? Do you know what I mean?
I think I know what you're saying.
Okay.
So because it's like the polymerase is still present, but it's just unable to fully do its job, fully bind.
So to follow up on that, do you expect phasing to come from a stalled polymerase or not?
Not really.
No, 'cause it's not active.
And in the elongating one, they're phasing more because it's like actively going through the process of transcribing. So they're moving around.
That's not quite right. People have, so that's okay, failures. Other people wanna correct that. It's close, but it's not quite there. Unless-
Well, it was kind of like what's in our figure is that it shows that the nucleosome actually like shifted a little bit. Actually, yeah, so they shifted like a little bit, but then, there's this thing where like protein needs to come in and help get polymerase actually going. And so that's why it's not actually actively transcribing.
Do you need actually transcribing polymerases to get phasing of nucleosome? Okay.
Basically, the stalled Pol2 is still bound at that region, it's just not active. So like if it's still bound there, then that nucleosome will still come in position right after. So you should see that's.
When you look at figure 2, do you see phasing? In expressed versus not expressed?
You still see that phase one.
You went,
Don't they still phase, they just try to block it more safely and screw it.
that's true at some level, and also that the phasing is not as stark, right? Right, because in stalled, situations with stalled polymerase, you don't, not as many polymerases have, not as many promoters have the polymerase bound. But anytime there's a polymerase bound, there will be phasing. And what Levi said is true as well, like that what your figure is saying is that that phasing is that it starts at different places, but it's still phasing. Right, so, yeah.
So when like the stalled, the pole two is bound but not yet actively transcribing, so it's the stalled pole two. Is it just the one nucleosome after it that is phased, or is it the rest of the strand too? Because I guess it's kind of going back to figure 2, but for B, there's only the one peak at +1?
Yeah. Yeah, that's all the resolution of their approach could see was the +1 nucleosome. But I expect that that's because that the vast majority of those promoters don't have a stalled polymerase, right? There's only a fraction of the non-expressed genes that actually have a stalled polymerase sitting on that. So I just think that the signal probably isn't large enough for you to be able to see the rest of them.
Okay.
So if there's a polymerase there is phasing. Right? And by phasing, we mean ordered nucleosomes around that promoter region, right? Because if there's something that's stuck on a promoter, those nucleosomes will have to order around that polymerase. If there's nothing there, then they can go random wherever they want to go. Right? But if you have something that's set, it has to structure around that order. What else? Other questions from the group?
Okay, so did I get this right? So like only for the stall, was that the one that only has like a fraction of the nucleosomes that actually has a polymerase by them? Or like bound in them?
Well, based on what you were saying, what I got is that with the stall, some of them do have it, but not as many as the elongated, so that's why it's so random. There's little spikes everywhere.
Yeah, so 3 is a follow-up from 2, right? Figure 2, they noticed that they couldn't see as much phasing in the B, in the unexpressed genes. So then they said, all right, well, let's distinguish them more, not just expressed, unexpressed. Let's then make 3 categories: elongating, stalled, and no polymerase. Right? So they're making now distinguishing And that's what A does. They said, okay, A, these transcripts are showing elongating polymerase, B is showing they're stalled, and C is showing that they're just not there at all. And then they use those categories to look at E, C, and D, and whether nucleosome position is a function of polymerase being there or not. And that D is the clinch, right? No polymerase, no phasing. Actively transcribing. And in that case, what does C tell you about phasing? Can you show us how C shows us phasing?
It's in the dips, right? Like the different spikes, like there's this and that and that.
Yeah.
But they're not very uniform.
Right. The fact that we see signal in the spikes means that there's phasing. No phasing is what B looks like. It's just noise, which you guys explained well, right? So make sure that, I think Hannah said it, but it should make sense that there's nucleosomes, even if there's not polymerase. It's just that they're not ordered. They're across the population. They're all random places. When they're all random places, all that you see is static. Okay, so it's helpful, like, if 5 of us began to speak, recite some lyrics of some song, and we did it in unison, you would know what we were saying. But if we just randomly started at different times those lyrics, you would not hear any of the words, right? That's what we're talking about here, right? Our phasing of us saying those words together would produce very clear peaks. You would know the words, you would know the beat that we're off. But if we just start randomly, you're gonna get static. That's what D shows.
One question, like, and you might have said this already, but, like C versus B, C has a lot higher spikes, so it seems like there's more nucleosomes being found in B.
Yeah.
Isn't there like the same amount, or more is elongated?
Yeah, it's a good question. I'm not sure that I have a good answer for it, because if you look at the scale, the first thing you look at is, are the scales the same? And they are. And so then you say, well, that's strange, elongating polymerases, you would expect to have a stronger peak. But then you might argue, well, so let me, before I just put the answer out there, does anyone have a thought about why there's, let's go to that.
Could be that since there's like less organization, that there's like, this kind of happenstance that there was more at that one particular point, as like if you were to just throw them all in there, that this one spot had more compared to like the organized one where they're all kind of like.
You say organized, what are you referring to?
Like the nucleosome, sort of put in order with peaks and stuff.
Close. It's fuzzy there. So what's different between a stalled polymerase and an elongating polymerase that might cause the nucleosomes to be different?
There's more polymerase bound to the transcription start site, so you'll see more like phasing, the
Which
layers,
one?
for B, you're gonna see more like phasing because there's gonna be more, polymerase bound to the start site, and so then.
What Bailey is saying is that that's not true. Look at the peaks. She says that B actually you see less phasing because the peak is a lot smaller than what you see in C.
I was thinking that maybe because there's more polymerase there, the phasing is like, it's more uniform, like you see like a pattern where it's like, when you look at C, it seems like random, and then you see the start site, a little bit of phasing, and it looks a little more random again.
Yeah, that could be. So it doesn't explain why you see a smaller peak in B though. What might explain? Right, so we know that what causes phasing is the presence of polymerase. What might cause B to be less? Yeah.
Could it be because, there's a lot of genes with stalled POL2 and only a few of them are starting to actively transcribe?
So that could be, what they did, if you look at the methods, is they actually tried to normalize that. So they took the same amount for A, B, and C, but that's a good thought. Ends up not being true. All right, so here's one possible, I don't know this is the answer, but one possible scenario is that if it's elongating, what percentage of the polymerases are gonna be at the promoter in elongating? All of them, some of them, none of them?
some of them, so some of them are new.
There you go. Some of them won't be there. So if some of them aren't at the polymerase, or not at the start site, that will affect phasing, right? Because they're gonna be found, and that's what A showed. You see that long tail? The polymerase is spread across the gene, right? Because some of the polymerases is halfway through, some are three-quarter through, some are all the way through. So but in the stalled one, all of the polymerases are sitting at the promoter. So that's gonna create a greater amount of phasing because the whole population of polymerases is at one place, one time. Is that what you're trying to say?
Yeah, no.
Okay. I didn't catch that from your words, but that is great. I'm glad you're on the same page. Does that make sense? All right. 4. So this makes, I can put that happens.